Welcome everyone to the next IITE seminar, where it is my great pleasure to welcome Anuraag Bukkuri. I've met Anuraag first in the MME conference a few years ago, and I was very impressed by his talk. I also reviewed one of his papers, which I was also very impressed by. It was a big review of using evolutionary theory in the context of cancer, and I believe we will be hearing more about that today. So without further ado, Anuraag, the floor is yours.
Awesome. Well, thanks, Yuri. It was lovely meeting and running into you in many different contexts. And thanks for the invitation to speak here. I've obviously loved many of the talks I've attended in the past. I think it's a really cool seminar series that you have put together, and so I really appreciate that, and of course the invitation to speak here. So so I'm Anuraag Bukkuri. I'm a PhD candidate at the Moffitt Cancer Center in cancer biology. And so I want to talk to you guys a little bit today about modeling the role of the polyaneuploid cancer cell state on therapeutic resistance. And as you might expect, this talk will likely be more applied than most of the talks that you've heard before. But I hope selfishly, my goal at the end is to sort of convince you that biomedicine, and in particular cancer, is a really fertile ground for a lot of novel eco-evolutionary thinking and modeling. So like I said, I am in the Moffitt Cancer Center, which is a really unique environment. So I work right up here in the research building, and the patients are literally just a minute away. So we see them on basically a daily basis. And this has provided a really interesting setup and really sort of keeps Moffitt's mission, right, to contribute to the prevention and cure of cancer really to the fore of everything that we're doing. And so why is this important? Cancer is a massive public health crisis. So 10 million people die of cancer each year. And to sort of put that into context that I can, more easily understand, that means that around every 3 seconds, someone is dying from cancer. Um, in addition, it's a really big, economic burden for, across the entire world. So last year alone, 193 billion US dollars alone were spent just on cancer research. So this is not the clinic, this is not treatment, this is just on cancer research. And so where has that gotten us? We've had improved treatments for radiation therapy, new cool chemotherapeutic drugs, these novel immunotherapies, and new sort of surgical resection techniques. But unfortunately, over the last several decades, we haven't moved the needle basically at all on these advanced metastatic therapy-resistant cancers. And so you might ask, okay, why is that the case? We have a lot of smart people, a lot of money going in here. What's the issue? And I think the issue is really this gene obsession of a lot of cancer folks in the field. And so this is a quote from my favorite author, Edgar Allan Poe, in his short story, The Murders in a Rue Morgue. So this is a detective that's sort of questioning some of the methods used by the local police force when they're investigating this mysterious murder that happens. So he said he impaired his vision by holding the object too close. He might see perhaps 1 or 2 points with unusual clearness, but in doing so, he necessarily lost sight of the matter as a whole. And so I think this is a really apt characterization of a lot of the cancer research today, where everyone sort of has their favorite gene, favorite protein or pathway, and then they're sort of single-mindedly, obsessively studying this thing without necessarily thinking of the broader context, how this actually contributes to cancer and how that might help provide a cancer cure. And so with that, I sort of want to reframe our thinking from thinking of cancer as a disease of the genes to thinking of cancer as a disease of evolution. And as sort of I alluded to earlier, one of the main problems, really the primary cause for treatment failure, is therapeutic resistance. And so this is unfortunately all too common. So here a patient presents to the clinic with a very aggressive form of melanoma, a skin cancer. He's given a RAF inhibitor, which is a targeted therapy meant to kill all the cancer cells in his body, right? And 15 weeks later, he's all clear. There's no clinically detectable cancer in his body. He's sent home. He's happy. But 8 weeks later, he's back in the clinic because his disease is worse than ever. So this is obviously very jarring for the patient and as well for the healthcare provider because of this idea of therapeutic resistance. So we really want to hear about how we can solve this problem, or how at least we can understand this. And since we're in a theoretical ecology seminar, we're going to sort of think about how maybe mathematical modeling or eco-evolutionary thinking can help with this. And so when you give a therapeutic stressor or any sort of stressor to a population of cancer cells, there are two main pathways that the cancer cells can sort of take to adapt to the stressor. So one is evolution. So this is sort of genetic evolution. Heritable changes are passed down from generation to generation. or acclimatization, right? So this is where cells can acclimate to the drug within its own generation. So this could be behavioral changes, there could be transitions in cell states, maybe they move to a more favorable environment. But these are the two main ways they can adapt. And so we want to see how these interact with each other to promote therapeutic resistance. So first, evolution, this is the easy one. How can we model evolution? There are many ways to do this. One way to do this is using the G function framework, which I think, as Chris likes to say, is sort of the US version of adaptive dynamics, so very closely related. So we just have a set of coupled ODEs. So we have population dynamics and then evolutionary dynamics. So the population dynamics of some morph over time is just given as the product of the current population size, and then what's called this G function or fitness generating function, which captures the per capita growth rate as a function of an individual cell strategy, and then the population densities and traits of all the morphs in the population. And the trait or evolutionary dynamics is sort of given by Fisher's fundamental theorem as a product of evolvability, which is the capacity to generate heritable variation that can be acted upon by natural selection. And then the selection gradient, which basically just says, how does the fitness of a cell change if it changes its strategy a little bit? And so we can sort of intuitively visualize this on what's called an adaptive landscape, where we're basically just plotting the fitness of a cell as a function of its trait value. Here, we're showing a static landscape at one snapshot in time, but in reality, this is dynamically changing as the population evolves and the densities change and whatnot. So if you're a cancer cell at this yellow circle here, you're just going to want to evolve up this adaptive landscape in sort of this hill climbing process to reach a local peak here denoted by the green star at A. And we sort of created this really basic, simple web app pedagogical tool really here to sort of allow people who are not necessarily familiar with the math to play around and get a bit of a feeling for how these models work, again, without needing to do any of the modeling themselves. So that's all well and good. How can we deal with the acclimatization, the plasticity aspect of this, right? So that's a little bit trickier. So how do cells acclimate? Here, we're really thinking about how they can acclimate by entering a different cell state. So maybe they can go dormant. They can sort of fall asleep, or maybe they can become more mesenchymal. They can become more migratory, or they can enter, as we'll talk about later, a polyploid state. Right? So how do we think about modeling this? Well, if we want to include plasticity, we really have to take this life history or demographic perspective, which leads to this idea of state-structured populations, well known in a lot of the sort of eco-evolutionary world. Shripad gave a talk a couple of weeks ago touching on this as well. But why is this important? Well, for example, if you were sort of maybe a bit naive, you might think of a butterfly and caterpillar as entirely separate species, right? And you might model them as such. But you'll run into a lot of issues really quickly, especially in the evolutionary component, because obviously what happens in the caterpillar stage will impact what happens in the butterfly stage and vice versa. So we need some kind of unifying fitness function that will allow us to model the fitness, right, across these different stages in the population. And one way we can do this is using this sort of state structure modeling approach that we developed here, which is very closely related, again, to the other approaches in the field, where we can start with a life cycle graph, right, if we're sort of hardcore field biologists, or just a set of equations if we're a mathematician. And we can throw all that information into what's called this population projection matrix, and then use the spectral radius or bound of that matrix, which sort of controls the long-term dynamics as our G function, right? And then we can use the method that I described earlier to model what happens to the ecology and evolution and demography of the system. And so how is this actually useful in cancer, right? So we'll take a case study here, really focusing on the polyaneuploid cancer cell state. So what is the polyaneuploid cancer cell state? So polyaneuploid is really made up of two words, so polyploid and aneuploid. So polyploid really refers to a whole genome duplication, right, of a baseline genome. And aneuploidy is really common in cancer. Almost all cancer cells have some sort of aneuploidy, which really refers to sort of chromosomal aberrations, right? They might have extra duplications of certain chromosomes, or they've lost parts of chromosomes and whatnot. And so polyaneuploid then refers to this whole genome duplication of this baseline aneuploid genome. And so what are the characteristics of this polyaneuploid cancer cell, or PAC state? So they pause cell division and they're able to increase their genomic content in a process called endoreplication. So instead of undergoing cell cycle as normal, where you just go G1S, G2M, and then you divide, What happens here is actually after the S phase, after they've synthesized and replicated their genomic content, they can exit the cell cycle in G2 or early M phase, and then they can continue endocycling, duplicating their DNA content several times without ever actually dividing, right? And we think that this is sort of forms in a condition-dependent manner. So the more stressed cells are, the more frequently they enter the pack state. And so we've done some experiments to look at this. So in this upper right-hand corner, you can see, so these are prostate cancer cell lines. And you can see initially in the baseline conditions, they're all somewhat small cells, right? And you can see with these flow cytometry readings that they're all basically 2N cells. These are standard diploid cells with some chromosomal aberrations. But once you give this therapy, the cisplatin chemotherapy, you can see this drastic shift, right, in ploidy levels. So you see that they've increased to 4N or even past 4N, which means they've duplicated their genome several times. And you can see that the cells have become larger, right? But when you isolate those cells and allow them to recover, they go back to their pre-therapy sort of frequencies, right? So now you see again the recovery of this standard morphology. And you can see that down below a little bit more clear graphically with, again, the longer they're remaining in therapy, the more time they have to endocycle and become larger and have a lot of this genomic content. It turns out that these giant cells have been noticed in the literature for centuries. So we see them in even hand-drawn illustrations from Muller and Virchow in the 1800s. But really, until now, they've been ignored. They've been assumed to be destined for mitotic catastrophe, right? So they assume that these cells are just going to end up dying or senescing. They're not really contributing to any form of recurrence. They do notice that they are condition dependent. So they've assumed that they are somehow associated with cellular stress. But again, they're not really causative factor for recurrence. But here I want to show you a little bit of some evidence that this is not actually the case. And so this is going to be sort of an IncuCyte live cell imaging video that you can see, where all of these cells that you see here have been tagged with these Fucci reporters. And so when they're in the G1 phase, they're going to be red. When they're in the S phase, they're going to be yellow. And then when they're in the sort of late S G2M phase, they're going to be green. And you're going to be able to really clearly see these cells endocycling. So just pay attention to the cell in sort of the middle of the screen here. So as you can see, it's green right now, right? And then it's going to become red here when it goes to G1. Again, so it hasn't divided. It's just gone directly to G1. And you can see slowly it's going to be synthesizing its DNA. It's turned yellow now, and then it's turned green, right? And then it's going to endocycle again. So it's going to go directly into that G1 phase. But as you can see, the cell is still intact. It has not divided, all right? So there we go. There it's turned to the red phase. Again, still becoming bigger and bigger, duplicating its DNA content, but critically, it has not divided, right? And then, so what happens? What is the fate of these large polyaneuploid cancer cells? So what we see actually is that they can go on and seed the recurrence of the tumor. So if you pay attention in the center right here, you see those two little cells that are sort of fused together. You can see that eventually those cells will go on to actually divide and be viable. So they've split here. They're going to actually refuse for a little bit right after. But then you can see eventually they do split, and then they all go on to sort of seed recurrence in that plate. And so that's what we know. So how can we create a model of this? So again, it's sometimes easiest to start with this basic life cycle graph. So we have two states. We have our 2N state, right? Our aneuploid state. This is the normal cell state for cancer cells. And then we have the pack state. And so 2N cells can divide. They can breed true, but pack cells cannot. So 2N cells, they divide, they transition to the pack state. In order for pack cells to divide, they have to depolyploidize back into this 2N state. So that's what we know. We've also assumed that the pack cells are entirely resistant to therapy. This is what we sort of see in the lab, whereas 2N cells can be sensitive. So basically what we've assumed is that the 2N cells grow in a logistic manner. There are constant background transitions between both states, shown in blue here. And then we have this drug-induced death term, right? So the 2N cells are the ones dying, and they also have this facultative transition to the pack state. So all that means is that the more stressed these 2N cells are, the more they're going to transition into this pack stage. And then again, we have our evolutionary dynamics as sort of given before. And how does that impact the population? That V trait comes right in here in that drug-induced death term. So as the population evolves resistance, that this denominator becomes larger, and then the drug-induced death term goes down, and the transitions to the pack state also go down. So this is sort of the model we created. We sort of use a strong inference approach to sort of generate several hypotheses on how the pack state can actually contribute to the evolution of resistance. And so I'll just show one here, which is the one we thought was most promising, this idea of evolutionary triage, in which the evolvability is dependent on the frequency of packs in the population. So the more packs there are, the more genomic content they have, so the higher the evolvability is going to be. And so here, these are just a couple of basic simulations showing what might happen. So the ecological dynamics of the 2N cells are shown in blue. The dynamics of the pack cells are shown in red. And then the drug resistance is tracked in black here at the bottom. And so periods of no therapy are in white and periods of therapy are going to be in green. And so as you might expect, before therapy, the 2N cells basically occupy the vast majority of the population. This is definitely what we see. When therapy hits, that population comes crashing down. You see this jump in the pack state. And then as the cells evolve resistance, you see this gradual decline in the pack state, right? And you see the same with intermittent therapy, really here just to show how the drug resistance evolves. So at the beginning, it evolves much more resistance because, again, the selection pressure is much higher. During periods of no therapy, there's no resistance going on, and then it evolves less and less as sort of time progresses. So this is great. Obviously, now we understand how the pack state works, right? Turns out that's not the case. This is not what we observe. When we took these sort of observations back to our experimental colleagues, this is what they said. So they said, look, we do see that rapid transition into the pack state. But what happens is that the cells tend to remain in that pack state for quite some time. And then they depolyploidize into their 2N progeny, but those progeny are immediately resistant to therapy. So there's no creative destruction of cells that enter the 2N state, right? So then we had to say, okay, we have to come up with a different hypothesis for what's going on. And we proposed this idea of self-epigenetic modification, right? So we thought, okay, via reverse transcriptional inheritance, the changes that a cell can undergo during its lifetime are somehow written back into its DNA and passed on to offspring. So sort of this Waddingtonian genetic assimilation going on, where sort of the phenotype that's produced in response to some environmental condition somehow becomes genetically encoded. And these are things that we're now also testing in the clinic, and we have some promising sort of sequencing results that show that this might actually be going on. But a lot of the evolutionary models are obviously not necessarily well suited to account for this. So what we proposed here is that we thought that, okay, if cells are sort of at the peak of their adaptive landscape, they're doing well, they're really happy, not much evolution is going on. If they're close to the peak, right, they're sort of near or within their fundamental niche, then we think, okay, the cells are going to evolve via evolutionary triage as normal. But when they're really far from their fundamental niche, we think this sort of self-epigenetic modification is going to kick in. It's going to allow those cells to jump up the fitness landscape and then sort of evolve via evolutionary triage. So how can we actually simulate this? We sort of use this Gillespie eco-evo-demo simulation. We switched to a sort of stochastic simulation framework just because it gave us a little bit more control over what's going on in that pack state. So basically we have birth, death, switching events, and then we've included sort of a mutational kernel. The cells can mutate with some probability when they divide. But the key aspect to really notice here is in green. So what this green part's saying is that, okay, if you're a cell in the pack state and you're going to depolyploidize, what you're going to do is you're actually going to assess how viable your strategy is going to be for your progeny. And so if you think, depending on some threshold, if you think your progeny is going to be viable, you'll go ahead and depolyploidize, you're going to generate that progeny. But if it's not viable, you will remain hunkered down in the pack state until you find a better solution. So again, this is sort of our workflow. Before we started with our ODE, we can go into our matrix modeling sort of framework, compute the spectral bound, and then have our deterministic simulation. Now we're just doing this sort of eco-evolutionary stochastic simulation. So we have that switching sort of process. We have a mutational kernel that comes in, and then we can run this sort of stochastic simulation. So how does this look? Evolutionary triage, very similar to before, basically the exact same. Subject modification, this is where it really gets interesting, right? So before therapy is administered, same dynamics, 2N cells shoot up, they occupy the vast majority of the population. When therapy hits, that population crashes, and then we see the pack state rise, and then this is what we were trying to capture, right? That stable pack state. So the cells are staying in that packed state. They're trying to figure out a solution to that stressor. And when they do, they depolyploidize, and then you see that evolutionary triage process commence. And then you can critically see that sort of paralleled in the evolutionary dynamics here with that jump in evolution, right? And so that allows them to really survive therapy, these really high doses of therapy. So then the question is, okay, if it's so great, if they can survive all the time like this, why aren't they doing this all the time? And it turns out that that's mostly because of density-dependent sort of competitive factors, right? So we ran these sort of in silico competition experiments where you had a cancer cell population that evolves via evolutionary triage and a cancer cell population that evolves via self-genetic modification, and you sort of compete them together and see what happens. And so we have sort of three main regimes here. So the gray regime is when they're actually sort of operating identically based on the self-genetic modification threshold that we set. So in that case, there's really no difference. But when you're in the sea green state, this medium stressor state, right? You are stressed by therapy, but you also have to worry about these density dependent factors. And so here you notice that self-genetic modification, the populations that evolve via SGM are the ones going extinct the majority of the time. And again, that's because when you're in that packed state, you can't divide. So you've sort of lost that competitive advantage, whereas in evolutionary triage, you can. When you look at the high stress state, that trend reverses because now all you need to do is survive. And so then self-genetic modification really has the advantage there. And again, these are things that we're now trying to test in the lab. We had another really interesting observation of sort of what's called the Baldwin effect. So basically, this is an idea where an organism or cell's ability to learn new behaviors and acclimate to these therapeutic stressors can affect its reproductive success and can actually be selected for. And so what I mean by this is that if you have, again, here we've looked at prostate cell lines that have been treated with cisplatin, so one drug, right? You take those, you run those through, they transition into a packed state, they transition back out of it, and you take those cells and you expose them to a completely different therapy, right? They do significantly better than prostate cell lines that have not been exposed to that therapy at all. And one way that we hypothesize that this might be happening is this idea of packed memory. So wherein sort of cells that have been exposed or that have been transitioned into the packed state due to some therapeutic stressor now sort of have a memory of that state and can reaccess it more efficiently upon subsequent therapeutic insult. And so this is also something that we have sort of ongoing experiments that we're trying to test. But a lot of this modeling really sort of gave light and suggested that life history enlightened therapeutic measures might actually be really useful. So rather than just trying to use that single chemotherapy like a lot of adaptive therapies do, you just use that single chemotherapy and you try to just toggle the timing of it, we say, what if we can actually use drugs that block transitions into the packed state and use that in conjunction with chemotherapy? So administer the drug, it blocks the cells from entering that really keystone state that seeds recurrence, and then you give the therapy, that might lead to better therapy outcomes. And so that's great. That theory obviously works out. We can bake in whatever we want, but does that really work in the real world, right? That's the real question. So I took that into the lab and says, asked that same question. So here we first confirm that this polyaneuploid transition does actually happen here again. So again, we've seeded the cells, we've dosed them, and we can see this really large increase in cell size, in their mass as well. You can see that visually, morphologically, and also in these sort of histogram plots that really you can see that transition into those high ploidy levels. Molecularly, we also sort of showed that PRB here is actually ablated when cells are surviving, when they've entered that packed state. So normally what happens is this PRB binds this E2F transcription factor, which is what's really required to start this DNA synthesis process out. So normally it binds this transcription factor, and then in the process of mitogenic signals, growth factors, cyclin CDKs phosphorylate PRB, which allows them to let go of E2F, which then can sort of allow S phase to proceed and DNA synthesis to occur. But here, again, PRB is ablated, which is what really allows E2F to begin the DNA synthesis, even without the needed growth factors. So this is really what's allowing these packs to continue to endocycle in the face of therapy. And we then looked sort of, did some sequencing, and we found a lot of genes and transcription factors that were involved in this process. And one that really stood out among others was AP1, which is really involved in a lot of differentiation processes, proliferation, apoptosis as well. And so then we came up with a pharmacological inhibitor of AP1. and we use that in conjunction with a chemotherapy like we intended to before. And we were actually able to show that that does lead to a significant decrease in the cancer cell population. So the cancer cell population hasn't been eradicated entirely because there are other transcription factors and genes that are important in this process, but it does, lead to a much lower tumor burden. And so this, again, this is sort of inspiring ongoing drug development efforts, and we're trying to use these life history alliance strategies and see if that can sort of help us here. So that I just want to come up with, mention just a few other short snippets that can sort of drive home the idea that modeling and biology can really be a two-way street, right? And so here in this work so far, we've assumed that cells are discrete, which is fairly accurate, right? Cells can either be in a 2N state, they can be in a polyploid state, but not all states are discrete. So this was joint work with Sophie Mullen and Stina Anderson at Lund University, who are sort of developmental biologists interested in neuroblastoma, which is one of the most common pediatric cancers. It's a brain cancer. And there are a couple of phenotypes, the adrenergic and mesenchymal phenotypes, that folks weren't really necessarily clear on whether they were cell types, whether they were cell states. They weren't sure what was going on. And so we were trying to do a combined sort of modeling experiment to disentangle that. But here they've sort of done these flow cytometry readings and shown that actually cells aren't actually just adrenergic or mesenchymal. They're sort of transitory cells, right? There's a continuum here, and this is seen across cancer with EMT transitions, macrophage polarization, and things like that. So then we had to come up with different ways to model continuously structured populations, and so we sort of shifted to an integral difference equation framework to do that, and this was sort of a paper that describes that process. And similarly, they noticed that size might be another structural classification, right? So cells could be simultaneously structured by cell state and cell size at the same time. So now we need to come up with ways that we can model populations that are structured in several ways. And so that's led to a few techniques that we can develop to help us understand the eco-evolutionary dynamics in those settings. And then finally, the last one we'll talk about are fluctuating environments. And so Chris Klausmeier is on this call. He's done a lot of awesome work here and shown how flow K theory could actually help us understand eco-evolutionary dynamics of structured populations in these fluctuating environments. And it turns out this is very critical in tumor biology, because cyclic hypoxia, these fluctuations in oxygen levels, are characteristic of the tumor environment. They're really key to a lot of cancer phenomena that we observe. And so, for example, here are some histology slides where they've looked at a couple of regions of interest, and they've really clearly shown that cyclic hypoxic fluctuations can even happen on the order of minutes, and then also on the order of days. And some of our own experiments have shown that those fluctuations can actually dramatically alter the response to therapy. And so it's important to understand how the eco-evolutionary dynamics change in these fluctuating environments. So then if a physician is then treated with a patient, is confronted with a patient who he needs to administer treatment to, he can sort of personalize the treatment depending on the levels of cyclic hypoxia, for example, in that patient's tumor. And so I'll sort of summarize there. So I think it's important. Genes are obviously important. They've helped out even in a lot of our studies here, but they aren't everything. We sort of need to take a little bit of a broader perspective to understand what cancer is, how we can really treat cancer. I think the old adage, nothing in biology makes sense except in the light of evolution, is critical. The evolution of play takes place in the ecological theater. I want to add my own sort of with demographic actors. So it's important to also consider these cell state transitions, which are often sort of missed in a lot of the cancer modeling work. I think it's really critical for math modeling and experimental studies to be a two-way street. So not necessarily just data fitting, but really sort of inspiring future experiments and new math modeling and techniques. I think cancer is also, I hope I've convinced you, a great model organism for a lot of novel eco-evolutionary thinking and modeling, right? And so there's a lot of aspects. So for example, structured population modeling in cancer is basically never done at all. And so there's a lot of obviously fertile playing ground for that. And there's still, ecology and evolution has a rich toolbox that I think cancer folks can use a lot. And so with that, I just want to thank my funding sources and my collaborators here. So Joel and Bob Austin are sort of the theoretical folks involved in this. Sarah, Emma, Chris, Sophie, and Stina are the experimental folks, and Ken Pienta is our clinical collaborator. And with that, I'll thank you, and I can take any questions if you have them.
Thank you so much, Anurag, for that great talk. So if you have any questions, please raise your hand in the participant list and I will call on you as usual. Okay, while people are thinking, I can start and maybe with questions. So one thing that came to mind is that as a complete layman, I would imagine that in cancer, maybe more than in most systems, the ecological and evolutionary timescales are very intermingled. But correct me if that's actually, you think that's a complete misconception. But if they are, how much can one use this idea that the leading eigenvalue of the fitness generating projection matrix is the fitness, because that would be true if we were in quasi equilibrium, so that things have settled down, so that we're in a stable stage distribution, so that the dominant eigenvalue, or in your case, I guess leading, because you use differential equations, leading eigenvalues are actually governing the, you get the question. Yes.
Yeah, no, I think that's a great question. And that's still, sorry, it's
all good.
it's gone again. Now I can't see you. Oh, there you are. Yeah, so I think that's definitely a great question, things that we're trying to wrestle with. And so the G function framework, I think this is maybe like one of the only actual real differences that I see with the adaptive dynamics framework, is that it doesn't necessarily assume that timescale separation between the ecology and evolution, which makes some of the math more complicated. So in this paper, we lay out sort of conditions for convergence stability. And in the adaptive dynamics case, generally, again, you have this timescale separation. So you just have to consider the the evolutionary dynamics. But with ours, you also need to factor in how the ecological dynamics impact that sort of convergence stability. And so there are definitely different stability criteria that you can use, that you have to use when dealing with this framework. But I mean, the question that you raise is obviously a great one, and things that we're still trying to consider, because like you said, it's not at an equilibrium, right? When we're modeling this, and so we still need to come up with different ways to do that. I mean, the other thing is that we also have that stochastic framework, right? That we were mentioning, that can also help you get around some of those.
So I guess the question that comes up is this even relevant? Because of course, nothing is all ever at exactly equilibrium, duh. But
Right.
Are the conclusions, in your experience at least,
Yes.
if you have any, are the conclusions you draw from the idealized quasi-equilibrium assumption theory consistent with what you see from the Gillespie algorithm, or
Yes. Yeah.
Yes, they are. Okay.
They are. Yeah. Yeah.
Okay, that's very good to know.
Yeah. Yeah. Yeah.
Okay. Yes, thanks. Jessica, please go ahead.
Sorry. Hi. Hi, Anuraag. How are you?
Hey, friend, good. How are you?
It's been a long time. This came about through like a completely different way of how I heard about that this talk was happening, and it did not come from Moffitt in any way, shape, or form. So I felt like it was kismet, and I thought that I would show up. I am an old colleague of Anuraag's. I worked at Moffitt in the IMO for years, and this is so cool to see how this turned out. I kind of left a while ago, and this is just delightful. So congratulations, Anuraag. First off, just amazing. The only question that I have, and it's not going to be a theoretical question because I'm annoying like that. So when these cells go into this pack state, right, and it's this this constant pack state, this one that's not really freaking out, and this is where you're saying that these cells are in this state and that they're waiting to come up with the solution to the problem. How do they test that? Okay, so they're coming up with ideas, right, which is usually some sort of like antigen presentation, slowing down of some drug efflux pump, I don't know, right? Like there's a million ways that you can become resistant to something, but you generally have to try it, right? So if you're like coming up with these ideas, how do they like come out of the cave and like try it without dying? Because this is kind of assuming that is it assuming that a single pack cell is coming up with the solution, or is it a cohort of pack cells that are coming up with solutions, trying, dying, and eventually one of them gets it. So is it on a population level, or is it truly a single pack cell? And how, if it's a single one, that I don't get it. So that's a long story. Now you can talk. I'm done.
No, that's a really terrific question, and we are still trying to understand how that happens, because I guess so I can mention that the competing hypothesis is sort of what you mentioned, in that is that selection is actually happening to the cells in the pack state itself, right? So that is one of the competing hypotheses. We think there's evidence for both. There still does seem to be some subject modification, but we have no idea. The much smarter biologists have no idea either. We have no idea how this is happening. We're doing sequencing, so we're sort of sequencing the cells, the sort of 2N cells, and then the packs and then their progeny, and we're trying to see where the differences actually are, and then to see are they genetic, are they epigenetic, but we still, that's still stuff that we have no idea. In order to sort of get around your question, to sort of explain to biologists that we really have no idea, I sort of tried to call them ghost progeny. So I said that they produced these ghost progeny, and somehow these ghost progeny were tested, because we really have no clue. We have no clue how this is happening.
It's such a wild thing. It's such a wild,
I think you mentioned it correctly, but I mean, in biomedicine, I mean, the only really times that it's really proposed subject modification is sort of VDJ recombination and sort of the antigen presentation system.
right.
That's basically it. So we're still, it's a really exciting question thing that, it would be really cool to understand how that happens, but we don't know yet.
Yeah, I feel like I'm always a big fan of using like the theoretical stuff to make people think about it differently, right? Like there's the antigen presentation idea, but really, like, is that really what we're going with, right? Like there's got to be something else.
That's exactly,
yeah. So this is nice because it's like an actual concrete example of like, well, here's a point that we don't understand. Let's go find some experiments. Are you running? Oh, you're doing the single cell sequencing to see where the change happens.
That's right.
It's super interesting. Well, thank you again. I'm so glad that I popped in for this.
Thanks.
It's great.
Definitely. Yeah, let's catch up sometime.
Yeah, definitely. Great talk.
Thanks.
Thanks so much, Jessica. Chrissy, please go ahead.
Okay. Hi. I was wondering if you could talk a little bit more about where density dependence shows up in your models. I think you mentioned that there are some density dependent regulation processes going on. and I think you were talking about it in the figures where you have like anyway, that's basically my question.
Basically just in the logistic growth is where it's really coming in. And so then what's happening is that cells that are evolving via cell subject modification are stuck in this X2, this pack state. So they're not able to divide at all, because this transition to a 2N state is gone too. So they're just remaining in that pack state, whereas the other population is able to basically fill up that entire carrying capacity, is what's happening.
So then can you say again, like, how does that then influence the like cycling and the evolution of resistance that you see from this modeling framework?
Oh, so when I was talking about that competition, that was mostly trying to explain why cells don't always undergo subject modification if it's such a great idea. And that was because, again, there's that density dependent effect, so that if cells aren't that stressed, it's not really great evolutionarily to just stay hunkered down in that pack state.
Got it. Okay. Thank you.
Yeah.
Thanks so much. Are there other questions? I have a semi-related question, maybe, which is when you do the Gillespie, you have to make a choice as for what goes into birth and what goes into death. Have you tried multiple things? Does it even matter, and how was it partitioned?
So I think the main thing with that is really what happens to logistic growth.
Exactly.
So we have switching rates for all the facultative and obligate transitions, and then the drug is pretty obvious. So the only decision you have to make is what do you do with that logistic growth term? I think I threw that into the death.
Into the death. Okay.
Into the death. Yeah.
And then what's the birth?
The birth was just your RX.
I see that you split up the logistic into the just exponential growth minus.
Yeah, that's right.
Okay, I see.
Exactly. Yeah.
Thanks.
But that's a great point. Yeah.
Thank you. Any other questions? Oh yeah, Jonas, please go ahead.
Oh yeah, hi. Thanks for a very interesting talk. So maybe I just can't read the equations fast enough here, but it looks like the trait V, some kind of drug resistance trait, but it doesn't seem to be any trade-off for it really. Like it's purely beneficial. Is that right? And if
In
that's so,
this
is that
case,
something? If that's so, do you think like that is missing something, or is this like a good description of what is actually happening? I know nothing about cancer, so this is a question of pure curiosity.
yeah, so in some cases it does, there is a trade-off, there is a cost. What we say is there's a cost to drug resistance, but in many cases there isn't really a cost associated with it at all. So yeah, I think that's probably where I'll leave it. So yeah.
Thank you. Are there any other questions? I can ask one more random question, which is if one thinks, first of all, I just love it, and it's almost dark magic in the good sense, if that even makes sense, to have these one-dimensional evolutionary models tell us so much about about cancer evolution. Do you see potential for multi-dimensional trait spaces? Have you done that maybe? And if so, how are all these results messed up by having to consider, say, the genetic covariance matrix, which will rotate the direction of evolution in ways that might be very difficult to model?
Yeah, that's a great question. And I think there's only been sort of, yeah, I mean, in the theoretical evolutionary ecology space and this G function adaptive dynamics, there still needs to be a lot of work to try to understand convergence and sort of multi-trait systems and G matrix evolution. People still have no idea really what's going on there. So there's a lot of things. So in our thing, we haven't looked at that covariance matrix in the structured population setting. In the unstructured population setting, we have, and that leads to really interesting questions of double bind therapies. So that's basically the idea that you can evolve. So where it actually came out, my advisor, as you know, was Joel, who's a squirrel ecologist. So he gives the squirrel analogy. That's not actually where it started. So it started with this idea of family therapy. And so basically what it was is that if you have a kid and you have two parents that are telling you to do opposite things, then you're stuck in this double bind. Because if you do one thing, the other parent gets mad at you. And if you do the other thing, the other parent gets mad at you. And so it's a similar thing with the cancer cells, where basically we can apply one selection pressure to steer them into one phenotype and then target them with the other, right, with the other evolutionary sort of strategy. And so that's shown promise, and we've shown that in glucose transport, glucose deprivation settings, in angiogenesis, and even in sort of thinking more carefully about how biomarkers work. And so all those things, it plays a really critical role there.
Thanks so much.
Yeah.
Okay, are there any more questions? I guess not. So then, thank you again, Anuraag, for the great talk and great discussion. And I hope you will be in time to hop into your next meeting then. And thanks again for coming, both to you and to everybody else. And we will be in touch with the next talk that should be coming up in 2 weeks, I believe. But we
Yes,
will.
2 weeks, Mark McPeak.
Yes. Sorry? Oh, yeah, Mark McPeak.
Mark
Yes.
McPeak.
Fantastic. Okay, so until then, have a great rest of your day and see you next time. Bye, everyone.
Thanks.
Thank you.
Thanks, guys.